We're entering into an era where life itself is editable. One company has already brought back the genetic code of extinct animals. They've resurrected DNA that hasn't walked the earth in over 10,000 years. Not as science fiction, not as metaphor, as living organisms, engineered, grown, and born. Their founder isn't a biologist, he's a tech entrepreneur, and his team is using the same tools, machine learning, CRISPR, synthetic biology, to build something we've never had before, the power to rewrite evolution. Today's guest is Ben Lamm, the CEO of Colossal Biosciences. His company just unveiled the world's first genetically engineered direwolves. But this conversation isn't really about animals. It's about what happens when the interface to biology becomes a computer. It's about what we design next. I bring you Ben Lamm. Life seems so complex, so mysterious. How is it possible that we can manually edit it? Is there a specific breakthrough that's made it possible for you guys to de-extinct animals?
If you look at the DNA twisted ladder that everyone thinks of when they think of DNA, to be able to edit one individual rung and either knock something out or eventually even to change those into a different letter. So
eventually, we can't do that now?
Well, we can do that now. But when CRISPR first was discovered,
Right.
It then moved into single nucleotide editing where you could edit certain individual letters, right? And then it came into the DNA synthesis where we can actually synthesize big blocks and swap them in.
What do you mean synthesize?
So we can actually we do it internally at Colossal. But we also have third-party partners that we work with where we can actually string a bunch of those letters together. So instead of us wanting to go in and make like 20 edits or 100 edits or even 1,000 edits in a particular order, it may be easier just to sometimes synthesize that block.
So this block does this.
This block is this letters in this order. And so instead of us changing each one of those letters on that kind of twisted ladder, we can actually just design and build those letters in the right order. So we don't have to change them or swap them. We can literally just knock out that same little block and insert a block with the letters we want in the right order. So
okay,
DNA,
hold on. This is so crazy.
Yeah,
so
I love this is where we jumped in, right? So this is great.
When I say that I'm literally obsessed with how far this can take us,
Yeah.
I'm obsessed, but I like to actually understand
I don't
things.
even think that we're at the doorstep, like where we are, right? Because if you look at even what we're doing at Colossal right now, we use a combination of tools, right? So we're not using just one tool. It's not just like a knockout, meaning we're knocking out pieces of the genome. We're not knocking in pieces of the genome where we have something, we're trying to swap it out. We're not editing just individual nucleotides, like making individual letter changes. And we're not just doing DNA synthesis. We're doing all of it. So we're combining all of it. And that's what's called multiplex editing. And so one of the things that Colossal is trying to pioneer and push forward is this ability to take these tools that individually work okay, in some cases great, but a lot of times okay, and deliver it in a mechanism where we can make a lot of edits to the genome at the exact same time. And we're still we're running victory laps on the fact that we made 20 edits with one multiplex array, right? Which is unheard of.
Do you have to do that in one block, or you can literally do 20 separate, randomly placed
all
on
over the genome? Yeah.
Yo.
Which is amazing. So we announced this woolly mouse. So to give you context, we announced this woolly mouse, which is objectively cute, regardless of what you think about de-extinction. We made this woolly mouse. It's amazing, and it had 8 edits to 7 genes. So it just had 8 single nucleotide edits, right? So it wasn't like we weren't synthesizing a big block. We were literally making 8 specific edits in these 7 genes. But what was interesting is they were the same mouse equivalents of what we saw in our mammoth to Asian elephant research. So we do a lot of computational analysis. We spend a lot of time looking at ancient genomes, comparing them to their closest living relatives, understanding the difference, and based on the region it's in, it kind of gives us a clue of whether it'll make a change to the hair phenotype, fat metabolism, or whatnot. But these 8 edits, just these 8 edits, conferred all of the hair type, hair length, coat color, hair densities. Our chief science officer calls it floofiness, but basically how the hairs grow in slightly different ways. So it kind of gives it that kind of woolly texture. And then one was around fat metabolism. And if you look at like the control mouse, which just looks like your average smooth, like gray mouse, compared to this, our woolly mice look like, that they have a really cool fur coat on, right? And we're still testing whether the fat metabolism works. We don't know if that one, confers cold tolerance in the mice. But what's interesting about that is a lot of people were like, whoa, that's absolutely amazing. But it's only 8 edits. It's literally only eight edits. And people had made 8 edits before, but they had done them sequentially, right? So they made an edit in this line of mouse. That mouse, had offspring. Then they made an edit in the next line, and the next line, and the next line, and they stacked them over time.
So 8 kids deep. You finally have very different.
Yes.
How are you doing that? Is this writing on the back of a virus that knows that it needs to go in and clip? Like, I have a vague understanding of how CRISPR works.
You've got it right. So we design these CRISPR guides.
And a CRISPR guide is a virus.
So we go in, it actually delivers it, knows exactly where to cut it. But it's not 100%, right? So that's part of the problem.
Meaning what? It sometimes cuts in the wrong place?
It cuts in the wrong places. Sometimes there's a lot of what's called linear repeats in the genomes, meaning that there's places where there's the same letters, and some of these tools aren't quite there yet. I mean, we're still in the early stages. Like I said, I think we're just barely opening the door.
So it's like opening a book, and there's a few times where it says, and the people went.
Yeah.
And so if you tell it to look for, and the people went, you might be in the wrong chapter.
You might be in the wrong chapter or whatnot, right? And so, or if you changed it, it may forget people, right? Or may have knocked out people too. So there's these things called off-target effects where you get these unintended consequences, right? And so the way that we fix it, and the way that, this isn't where I think the technology will exist long-term, but where it exists today is we do an intense amount of screening. So we screen all of the cells before we edit them, right? So we do sequencing on all of them. So we're very heavy on compute. That may not be a surprise to you given my background in software. But so we spend a lot of time on understanding the existing genomes. Then after we make edits, after we let those cells kind of go through a couple passages, meaning they divide, and we then sequence the cells that fully divide. Before we even go into things like the woolly mice, people are like, well, how did you get, all 36 of your mice that were born were healthy? It's like, well, because we screened all those embryos. We didn't take any embryos to term.
What are you looking for? Because when I think about human embryos and screening like that, when people are doing IVF,
they're looking at morphological for a lot of times.
They're just reading the code in the DNA and going, we know what this means. Ooh, this means something bad. And so we're going to
I just went through IVF. So, and so we actually went through it, and I actually have a gene mutation that causes a truncated protein on the Titan gene. And so we didn't want to pass that on.
Which has already been mapped. People already know about this.
Which is crazy.
How much do we already know about this stuff?
I'll get pushback on this, but I think that we don't know very much. I think we are learning at an incredible rate, right? There's a ton of literature out there, but things like large language models and being able to ingest a lot of research, a lot of times people will call things different things in research. So if they're looking at a mouse model, they'll say, oh, this gene could have this effect, but that effect may be described differently in this paper than in this paper, right? So I think though there's been a lot of raw data created. I think that the kind of access to compute, AI, and eventually quantum, which I hear is only 2 years away, every 2 years. I think that those things coming together, we'll be able to ingest a lot of this historical data and existing data. I think that we are at this moment where, in the next 5 years, we will move from scientific research and scientific discovery to engineering. And I think that is
Say that in another way. Do you mean that instead of a bunch of people writing papers about this, we're going to know, okay, we've scanned 46 million people. This guy's 6'2", this guy's 5'4". Here's what it looks like. This guy has thick, luscious hair. This guy has really cheesy, thin hair. And we just start mapping all that stuff out, and we go, oh, okay, cool. I know these letters in this place are going to result in this characteristic.
Yeah, and I think we will, but that's what's called genotype to phenotype. That translation layer is pretty hard.
And that's not what you mean.
No, it is. So, but I think that's very hard. There are certain things so one of the biggest challenges that Colossal's working on is size. So there's not a single gene that you make a single edit in, and it makes and it's oh, well, we can now 3x something. We can now make a chicken that's 20 times larger, right? And that's great for farming.
Oh my God.
We're not there yet, right? We as humanity
We're literally going to have dinosaur chickens running around here in 20 years.
I'm not I'm not saying that, but
that was me.
We do get a lot of dinosaurs. That's the number one requested thing is dinosaurs.
God bless Jurassic Park.
Yeah, we get a lot. We've also heard that before too, surprisingly.
Shocking.
But I do think that we're at this point where we're going to start to understand two major categories. One is expression of genes into physical traits or phenotypes, right? So that we know that this gene or this combination of genes actually will have this effect, and it's highly predictable, right? So I think that is the world. So Colossal spends a lot of time in that genotype to phenotype mapping. That's like a large portion of our time. Secondly, though, I think that we're going to understand how, and there's been previous research to do this, but the application of these technologies to human healthcare, I think are going to be even more interesting than just like making woolly mammoths and whatnot. So the ability to understand that you have a predisposition to this type of cancer or this type of Alzheimer's or diabetes, unlike a single gene mutation that only requires a single edit, you have to be able to edit all of those genomes at the same time or all those genes at the exact same time with a high degree of efficiency, right? Because you don't want off-target effects, especially if you're dealing with humans.
Can you do this to somebody that's already alive?
You will be able to, right? And so what's great about Colossal is that we work we do a combination of editing in microinjection into embryos, but also in editing cells that then we use a process called somatic cell nuclear transfer, where we're taking the nucleus of an egg or of a somatic cell and putting it into that of a germ cell or an egg cell, also known as an egg cell. And so we aren't doing that, but a lot of the gene therapy work and a lot of these other works are looking at how we target different tissue types for delivery. So you've got the gene editing requirements, you've got the computational requirements, then you have the delivery requirements. I mean, for us, it's not as a high priority, but there's entire teams. I sat through a presentation at the Wyss Institute last year where there was like 3 teams that are just looking at kind of like air traffic control of how you push different targeted therapies to different tissue types in the body. So there are people working on it. Just
meaning we put it into the body, but we need to tell it exactly where to go and what to do.
Yeah. So it targets certain cells. Yeah.
Okay.
And people are looking at how that could be a form of treatment even for cancer, right? Like how do we target things better? So targeting is a huge thing in this field.
Targeting is a huge thing. That makes a lot of sense.
But it affects Colossal less, right? So we aren't focusing on much of it because like we're not trying to take a wolf and engineer in traits to it in a 3-year-old wolf. We're starting at the cellular stage and the embryo stage.
Okay, so you're taking advantage of the fact that viruses already do this. They go in and they snip.
Yep.
When you say that you're training a virus, how do you give it the sequence of letters or whatever to look for?
I think I told you this before. Is like I'm not a scientist, but I just get like nerd out on it. I come from a software perspective. So we actually have a teams that just do all of the different guide designs. So they actually design it. They actually put the letters in. They'll actually, in some cases, synthesize little pieces of the code that we want in the right order, and deliver that in the entire give that to the entire package that then goes into the cell to deliver it.
Okay, what's the craziest thing that you guys are doing right now with that technology?
The craziest, I'll give you 3 things that are just super weird, and I don't know if we'll get there, but we are trying to synthesize a massive block of DNA. There's a project that we're working on, without going into it, because
you can't, because Lord knows with that smile I want you to.
Yeah. So most people know we were working on the mammoth, thylacine, and dodo. People now know that we're working on, or we have worked on, direwolves. But there's other projects. But like, for example, with the
when you say, just for anybody that doesn't know you, when you say worked on direwolves, you mean they exist?
Yeah, they exist. So we have 3 direwolves right now. Yeah, which I'm sure we'll talk about, which are amazing. So Romulus, Remus, and then Khaleesi. So
crazy. They'd been extinct for 10,000 years?
10,000 years, a little over 10,000 years. We used a 73,000-year-old skull and a 12,000-year-old tooth to do all the computational analysis to kind of build
to get the
our
DNA.
genome. Yeah.
Right. Okay. Yeah. So craziest thing.
So 3 crazy things that we're doing right now. Number 1 is patterning both in the thylacine and in other species is really important, right? So like getting how the stripes, for example, we can't just like make up, like if we did the DNA analysis and saw that direwolves were striped, there's no preserved direwolves. So it's like, okay, well, the stripes don't have to be 100% from a fidelity perspective. But we know exactly what the thylacine looked like. There's pictures, there's color corrected videos, there's hides. So to get that right, we're actually synthesizing large blocks of DNA, larger than anyone's ever synthesized. And we're working to safely deliver that in the cell. And one of the technologies that we've developed is we built it really large and assembled it. We're then cutting it at specific points, putting it in, and then trying to stitch it together all at the same time. So that's one really insane hard thing that we're doing.
That's really hard because of the size of the block you're trying
Side
to
of the
do?
block, because it's pretty fragile, right? It's going into the cell. There's only so much.
So there's
you
like
can
a
push.
physical manipulation problem.
There's a yes, but it's all done with like chemicals. So we're not doing it with like tiny robots. That's
virus isn't? That's what I had in my head.
Yeah, but we aren't doing it, right? The system, the virus is doing this.
But does the virus literally carry it like a tugboat?
It carries it like a tugboat.
Get
cuts
out!
It
That's
and delivers
insane.
it. So that's one. A separate thing that I think's pretty interesting is we want to create a universal egg, which I know sounds a little weird, but because when you look at doing somatic cell nuclear transfer, in this one,
do tell. What's a somatic cell?
Okay, so a somatic cell is basically any cell in your body that's not a egg or sperm cell.
Got it.
So whether that's a tissue, we actually have started using what's called EPCs. We did this on the Direwolves, which was pretty cool, endothelial progenitor cells. And we love them because they're only partially differentiated. So if you think about like pluripotent stem cells or all these stem cells, they're in the most naive state so that you can use a bunch of different chemical factors to convince them to turn into different tissue types, right? And so you can use these transcription factors to convince them to turn into different tissue types, right? Which is pretty interesting. So we're using that with like hair. It's kind of creepy, but interesting. So in addition to the woolly mouse, we actually built hair organoids for mammoths. So we are growing hair follicles.
Just to make sure it works?
Oh yeah, to make sure it works, right? So making sure that the edits that we are making in our elephant cells will actually deliver the phenotypes that we're looking for, right?
Hold on. So we know enough about the soup it would need to be in to grow that we can grow the hair by itself?
So that's a whole nother thing. So if you look at, this is what's interesting about like why I'm super fascinated with what we're working on is we don't just have to get multiplex editing right. We don't just have to get computational analysis right. We don't just have to have the screening for off targets right, or the embryology right, or the somatic cell nuclear transfer, which we'll talk about, the cloning right. We actually have to do things like media conditions, like the soup, right? Like we have to get that right. And then we have to also create different soups or medias in order to make them change and in certain tissues do certain things.
Damn. So
it's not just editing. So you have to build the whole system. And so when, it's funny, like when we launched the woolly mouse, people were like, we have a lot of people that love us. We have some people that question us, which is great. I think it increases the likelihood of us having great conversations about science, which is awesome. But some of the people that have questioned us and they push back on us, one of the things is they're like, well, it's only 8 edits. And I was like, yeah, but it's 8 edits in this system. Like we're focusing on this, right? Where when in reality, we need to look at the whole system.
Yeah,
right?
I find anybody that says this isn't interesting completely uninteresting. Like I don't even get that line of
but it's
questioning.
Okay.
I hear you talk about it a lot. Like that one to me is just like, what? I don't have time for that.
But I think that I believe in personal choice and freedom, and as long as you're not hurting anyone. And I think that what's interesting about this is that people sometimes don't respect like the woolly mouse is like a technological triumph. I know it sounds insane because it's like
why
cute
would
little mouse.
That sound?
But it's amazing. We literally we took 60 genomes in varying lengths from a 1.2 million year old steppe mammoth all the way down to a 3,500 year old woolly mammoth. Built a kind of this pan-genome model, compared it to Asian elephants, African elephants. By the way, which genomes didn't exist. We had to go create those genomes. Like coming from software, I just assumed.
It just hadn't been mapped.
Yeah, no one had mapped. So no one had made the genomes, and then no one had annotated the genome. So no one had gone out and said, this is this region of the genome, this is this region of the genome. No one had done that. Coming from software, I just, and I knew that like 23andMe existed.
Right, there'd be a bank somewhere.
Yeah, I just thought like, oh, we'll just log on to the GCP of species and be done with this thing, right? Like we'll just write, comparison algorithms and call this a day. Yeah, but none of the data on either side had existed, which was crazy. So we had no data sets to start from a comparative side. So once we did all that and we identified all of these core regions, we started growing things like hair organoids. But before we grow hair organoids, we had to actually figure out how to turn elephant cells back into pluripotent stem cells so that we can then turn them from pluripotent stem cells, reprogramming them and turning them into organoids.
Jesus, man.
So there's a lot that goes into this. And, we've only been around for 3.5 years.
I was going to say, there's not only a lot that goes into it, but there's a lot you've already solved.
Yeah. And I mean, we're a long ways away from everything we want to do. But like that just little model right there, I think is like borderline magic. And so if you take all of that and then you map all of those edits that drive the hair phenotype, which is what we're looking for in our woolly mouse, to a mouse equivalent model, right, of that specific gene edit, because we didn't want to just put mammoth genes in a mouse. We thought that was unethical. And we thought that, there's 200 million years of genetic divergence between the two. So it probably wouldn't have been compatible with life. We actually had to engineer it. So we want to do this safely and responsibly. Then you sequence it, and all of that had to work to produce a mouse. And we produced 36, and they're all healthy.
Jesus.
And objectively cute.
They definitely are cute. There's no doubt about that. But like you said, I don't know that it's borderline magic. Sounds like it spills over into proper magic.
Yeah.
This is by far the most sci-fi episode I've ever done. This is bananas. Okay, so we know the soup. We know the areas of the gene that we can edit. We were going through the 3 craziest things. That's one, these gigantic blocks.
The second one was going back to that somatic cell nuclear transfer, where we actually take the nucleus of a cell, of a somatic cell, and put it into an egg cell. Well, you have to have egg cells. So you kind of have like 3 choices for egg cells. Choice A, or 4 choices. 1, you can harvest egg cells from animals, right? And do it in a humane way where you actually have to, in some cases, develop devices to we do this on the Northern White Rhino Project, where we actually have to go extract eggs so that we can biobank them and fertilize them so that we have them in case that species goes extinct, because it's functionally extinct. There's only 2 left.
And you'd rather start with a full-blown egg than like a skin cell.
You need a germ cell, so you need a germ cell. Secondly, you can make germ cells through a process called gametogenesis, because they're gametes. And how you do that is you have to take a cell, like a skin cell, turn it back into its most naive state, pluripotent stem cell, and then from there you actually have to reprogram it and get it to turn into an egg cell that you can then edit. Crazy. Right? And gametogenesis
when you reprogram it, what are you doing?
You're adding a combination of these chemical factors. Your
is this the
Amanaka factors, yeah.
Okay. Yeah. So it's basically a chemical sequence.
Yes.
That does this?
Yes.
I can't believe somebody figured this out. Okay.
Yeah, it's crazy, but they're all different for different species, right? And so Colossal works in non-model species, right? So like most people will study mice, pigs, non-human primates, right? And so we're working in wolf cells. We're working in fat-tailed Dunnart cells. We're working in pigeon cells. Like we're working in elephant cells. We're working in these cells that just don't get a lot of attention. And they just don't have a research. So we have to figure out those soups. We have to figure out the chemical transcription factors, the right combinations. And they all have uniquenesses, which is crazy. So closing out the 3 crazy things, and then because we can go on tangents for this forever because it's fun and interesting. But one of the things that we're trying to do is to make a universal egg. But the egg donor will have a mitochondria, right? So we all have these like little powerhouses of our cell, these mitochondria, and there is an ability, the further you go genetically, in time, that you could have mitochondrial rejection, meaning that the mitochondria in the egg may not be compatible with what's in the nucleus, right? And so I don't get that deep into the epigenetic side of it or in the mitochondrial rejection side of it. But one of the things that we're trying to do is if we could make a universal egg, that would be incredible for like all of conservation and obviously all of our projects, right? Because then you would never have to harvest oocytes or egg cells from a species, especially an endangered species.
When you say universal, do you mean any species?
Well, we're starting in mammals, right? So it'd be mammalian work to begin.
Is that all?
That's what we're working on right now.
Jesus.
Yeah. It's hard. Yeah.
Okay, so failure of imagination on my part. I never would have thought that that's even possible.
And we don't know if it is yet.
But the theory is that there is a point at which we all share a set of things.
The egg cells for these different species are so different, right? But if you could get the right housing structure, and if you could actually build the right ways to either change or manipulate the mitochondria or do mitochondrial swap in the egg cells, then you'd have kind of the building blocks, like the Legos for building a universal egg. So then, because there are species, so gametogenesis, taking cells back to their pluripotent stem state, their most naive state to then program from, reprogram from, is hard. It's very, very hard. Hasn't been done. It's not like, once again, there's not the GCP of that. There's not a book that just tells us how to do all this, right? Secondly, then going through the gametogenesis process of making eggs and sperm, that's insanely hard. That's a harder than making like a skin tissue. So if you could, not have to do that for every single species, and maybe AI and quantum just magically figures out, who knows? But I highly doubt it. If you could make a universal egg, or at least have the building blocks for a universal egg, when you're working a non-model species like the northern white rhinos or elephants, you could just use that. So that's the second thing that we're working on that's super weird. And then the third thing is the artificial womb. So we are working on a full end-to-end system to grow everything ex utero.
Yeah. We'll get back to the show in a moment, but first I want to talk about building wealth. Crypto ownership surged by 40% last year, but the real opportunity isn't just owning crypto, it's putting it in your retirement accounts. The problem is most financial institutions are moving at a glacial pace to offer this option. That's where iTrust Capital comes in. They've cracked the code on making crypto investing simple, secure, and most importantly, tax advantaged. iTrust Capital has built a system that's incredibly easy to use, backed by bank-level security and designed specifically for retirement accounts. You don't need to be a tech wizard or crypto expert to get started, just someone who wants to potentially supercharge their retirement strategy. Visit itrustcapital.com/impact and use code IMPACT when you sign up to fund your account to get a $100 bonus. Again, that's itrustcapital.com/impact and use code IMPACT. And for any financial professionals listening, iTrust Capital is now selectively partnering with forward-thinking institutions to bring this technology to your clients. If that's you, email partnerships@itrustcapital.com to learn more. This is a paid advertisement. And now, let's get back to the show. Tell me more about the artificial womb. That's like one of the, in my prep, I was like, okay, I've got to know how far along we are with that.
If we are successful by the end of 2025, or probably end of 2025, we'll show some implantation in a model species like a mouse. By the end of 2026,
When you say implantation, what do you mean?
So what's interesting about this is that the placenta is this like awesome, amazing thing, but it comes from the embryo. So we don't have to engineer that, right? We just have, I'm going to massively oversimplify it, but all we have to do is to create the right environment with the right chemical cueing in the right place. If it is a placental mammal and it needs to implant, like 99.9% of placental mammals, like a mouse, then we have to give it a uterine wall or a synthetic wall that it can implant into. So that means that
So you'll implant an artificial womb.
So we'll implant, I always thought
this was going to be like a plastic bag.
So we'll hatch. So I think though there's a part of it that is a plastic bag. That's the most outer component. But I do think that you need internal component. We're doing it with a combination of hydrogels. Like we're using these like super sticky membranes, but we're also using bioprinting. So we're actually having to print insanely small because it's massively vascularized, right? Because if you think about how it works in like a human body or in a mammal, the embryo will hatch out a placenta. The placenta will attach or implant into the uterine wall. That uterine wall, the placenta, which is almost like this awesome alien. So it's amazing.
Literally. The more you look at this, the more you're like, oh God.
Yeah. But what's great is like, I was like, we don't have to solve that part. Biology does that part for us. Like it does want to work and want to live. And so it has to then connect and invade into the placental wall, but it has to be able to, obviously stay implanted. So it's got to be sturdy enough, but it can't be too sturdy that it can't implant. It's then has to be able to be vascularized enough so that it's getting the right level of oxygen, nutrient, extramen exchange, and chemical cueing, because some of that chemical cueing comes through the placenta. Some of it also comes from the uterine wall in just that kind of amniotic sac. So you have to figure out, but once again, it sounds more like, to me, like a data problem and a tissue engineering problem. then it doesn't sound like magic. It sounds like it's just data. It's like if we track every single day of pregnancy and we test and understand, what that chemical cueing is, then kind of like the transcription factors for reprogramming stem cells, we kind of know what the variables are, right? Is it glucose? What are these different knobs? And then we just have to tune them for every different species. And I think over time, if you get enough data, then you can probably get 80% there. So if this goes great, by the end of 2026, I predict we will have the first mammal born fully ex utero, which isn't that long from now.
Not even a little bit. It's like 18 months.
Yeah, it's not going to be a mammoth, it's not going to be a wolf, but yeah.
Do you already have one in mind?
It'll be a mouse or a marsupial.
Interesting. Are they just higher turnover rate?
Yeah, there's our fat-tailed Dunnart, which is our model species.
Fat-tailed Dunnart.
Fat-tailed Dunnards. It's literally the cute. It will give the woolly mouse a run for its money on objectively cute factor. So, but if you look at if you look at them, they only have about a 13.5 day gestation because marsupials have so much gestation that happens outside the womb. Mice have a little north of 20 days.
Got it.
But mice, unlike mice, the placenta. Technically, marsupials are placental mammals, but the placenta only shows up for a cameo for like the last day of gestation. So it's not really needed in terms of like gestation for the fat-tailed Dunnart.
Right. Okay, so when you say something will be born ex utero, would I be able to walk into the lab and see it like in its placenta just growing?
One, if we achieve it, and then two, if we achieve it and it's highly replicatable, then yes, we would probably stage gate it. So you'd have it at every single, like you'd be able to say, oh, here's device 1 and it's at day 3. This is at day 7. This is at day 24, or in this case, day 20 and it's birthing.
Bro, this is so sci-fi.
Yeah, it's crazy.
Legitimately.
But I actually think that artificial wombs, if we're successful, will have way more impact to conservation than anything else. Like we work on the Northern White Rhino project, and our job is to help them.
Is it just because you can do so many?
Yeah, so you can do it at scale. That's the beauty of the artificial womb. If we're really good at this, it's great for animal welfare perspective, it's also great for conservation because, instead of having to do IVF in rhinos to help save the Northern White Rhino, right, and put them in Southern White Rhinos to birth them, this is how the technology stack immediately applies at scale to conservation. We could take a blood draw, isolate EPCs, endothelial progenitor cells, which is what we did on the wolves, edit those, engineer in genetic diversity, right, from a population map that we've done, based on like taking tissue samples of Northern White Rhinos in frozen zoos, in museums and other collections.
To overcome the cloning problem.
To overcome the genetic bottleneck problem. So then they're not all clones. They're all they have enough genetic diversity, so they all become their own founder lines, right?
Yeah.
So then you take that, then you do somatic cell nuclear transfer. You make an embryo, put it in a universal egg, or use gametogenesis to make it, and then you take those embryos after you've done somatic cell transfer, and you put it in a ex utero device or an artificial womb, and you grow it to term. So the system. Once again, the system model that we're applying to make mammoths or direwolves or thylacines or whatever, has a direct application where we could become a technology partner for governments and NGOs around the world to go take our technology into conservation at a scale we've never seen.
This is a world that I don't think people fully understand, like how transformational this is going to be. We have a lot more to cover, but I do want to touch on the ethics of this. So I, unfortunately, am the unhinged guy that wants to see this stuff like pushed to the absolute max.
Yep.
But for people who their toes curl at the thought of us playing God, how do you think about this?
Well, I think that we have a moral, I think that we have a moral and ethical responsibility to do it. So, because, we actually have developed all of these tools and technologies as a species. We've become the apex predator on this planet. When we started the company, it was forecasted that we're going to lose up to 15% of biodiversity between now and 2050. That was 3.5 years ago. It's now 50%. Those aren't our studies. Those are independent studies with.
Why is it going so much faster?
Well, because we're accelerating climate change, we're accelerating overfishing, overhunting, eradication of habitat, right? And so polluting, cutting down the rainforest, all of that is a form of playing God, right? If like we go into the rainforest and like slash it all down and burn it, well, that is a form of playing God. We are changing that environment, right? I would even argue that taking cholesterol medication on a micro level is a form of playing God, right? Because you weren't designed or engineered or got the random roll of the dice to have a specific, cholesterol problem, right? And so I think all of these choices are a form of playing God. And so I think that we have a moral obligation to do something that, with these technologies that can not only help and benefit human healthcare, but could help species.
Okay, talk to me about human healthcare. Is this going to be dear parents, everybody is going to have kids via IVF. Don't worry, it's going to become very cheap. It'll be de rigueur. Like everybody's going to be doing this. Don't worry, it's not just for the rich. But you're going to want to do that so that you can select.
Here's the beautiful thing for Colossal is we only work on animals and conservation.
I know you're going to dodge like that.
No, I'm not going to dodge. I will answer the question, but I'm saying Colossal takes the stance that we as a company aren't going to work on humans because we already get so many crazy conspiracy theories about the company. Yeah. Given that InQtel and others are investors, they're like, what are they doing? What is this secret programs they don't tell us about? But we're pretty transparent about what we're working on.
I was going to say the things you're talking about already are like
they're already weird enough. Like
weird.
I think people give us too much credit and like that we've got like crazy stuff,
even more crazy things, even hiding more crazy in the background.
Stuff. But what's interesting is for us is like we at Colossal draw that line, so we aren't going to make those decisions.
Why not? Like Ben Lamm, why do you not want to do it?
Well, I mean, so
it's just too big of a headache.
No, I mean, I think that the separation for Colossal is good between animals and humans. Like we won't even work on anything in the non-human primate world. So anything in the monkey world, we won't do, or ape world, we won't do any of that because like there was a species, which I don't think there's any DNA called Gigantopithecus, which was not King Kong, but much larger than our orangutans and apes. We wouldn't touch that project. We've made a decision that we're not going to work on anything because people get asked us about Neanderthal, right? And so they're like, oh, could you do a Neanderthal? I was like, it's just a slippery slope. So we've made the decision.
It's a slippery ethical slope because it's too human.
It's just a slippery. Yes, it's just too human. It's too close. There's enough problems for Colossal to solve without doing it. With that said, all the technologies that we develop that have an application to human healthcare, we will license out, we'll spin them out. So other people may use our technologies to eventually grow, humans in artificial wombs and whatnot, even though Colossal will not do that, right? But I do think that, other people, thank God it's not us, but other people will have to think about what is the line between making selections for the betterment of a society from a genetics perspective before you go all out Gattaca or you look like it, or it looks like eugenics, right? And so those are really hard things. Like I'm glad that I don't have to like make those decisions. But I will say I just had a kid and went through IVF and there's now, have you seen this whole genome sequencing stuff that's come out?
No.
So most people, when they go through IVF, they're great doctors out there will look at a morphological grade, like how is the embryo doing on day 1, day 2, day 3, day 4, whatever. And so how is it developing? How is it ready? And they a lot of times they'll grade them based on the morphological, like how does it look? How is it behaving?
An ideal embryo would look just like this,
right? Yep. Or be behaving like this at this stage of development. So that's one. Well, then they started saying, okay, well, let's look for specific things like extra chromosomes or extra things that could be
facts,
could be really bad, right? And so they started looking at those types of things, right, and at least giving that data to the people, right? This is an issue. A lot of times people have miscarriages because something was broken in the genetics of it and of the developing embryo, and it just wasn't compatible with life, even though it was developing on day 1, day 2, or day 3. Well, so now there's a new company. Full disclosure, I use them as a service. I was so impressed with what came out of it. I did invest in them. So I just always want to be transparent about my investments. But Orchid Health, which is pretty awesome. And what they do is they take a tiny little couple of cells from the outer ring, so not in the developing embryo, but in the outer ring. They do full genome sequencing. So they're not doing like 23andMe. They're doing like really deep sequencing.
Because 23andMe is too surface.
To surface, right? They didn't look for everything, right? So they looked for the core
That's
stuff.
why they went out of
but they didn't go. I think there's a lot of reasons they went out of business. But they went. They do the full genome sequencing, so they can look for if there's specific markers. Like I had this Titan gene mutation, and so we did not want to pass that on because there's risk, so that we could screen for that, right? Well,
so you literally check the embryos. This one does. This one doesn't. The one that does, don't implant.
Yeah, I mean, so you build. So I built a spreadsheet to and I built a relevance ranking.
I like your style.
System of it.
How many embryos did you have?
So we went through 2 rounds of IVF, and so we ended up getting. I think at the end we ended up we down selected down based on this kind of distributed kind of bell curve that we made in a spreadsheet, and we ended up having 9.
Whoa. Okay, so 9 that you personally were like, I'm going to give these a grade.
Before we even did the morphological rating with the thing. So the other thing that's interesting, so you're talking about
what
like
other things other than the Titan gene?
So there's also, and so this gets into, and so this goes back into your question about like, and once again, this isn't Colossal making these decisions. This is me personally with, with my family making these decisions.
Yep.
And we started looking at, they actually do this like polygenic risk score, which some people don't.
What's polygenic?
So they basically look at like, we do know that certain genes and certain mutations, in addition to environmental factors, could lead to higher diagnosis of early onset Alzheimer's or diabetes or certain types of cancers, right? So one of the things that ORCID does, which I found pretty interesting, is it kind of gives you like the bell curve where most people fall, and then it can tell you like this embryo fell here and it has this mutation falls here, right? And so, I think that, the easiest grade is like, if you're looking at this, what the technologies currently allow for, the easiest grade is, is it compatible with life? Does it look more morphologically right? Okay, are there major things that we want to screen for, like extra chromosomes or chromosomal anomalies? And then from there, you can look for specific things, like in the case of me looking for this Titan gene mutation that we didn't want to pass on. And then it goes into this like area that's, I wouldn't say it's gray, but it's not like if you have this exact gene variant, you will get Alzheimer's.
It's off-white.
It's off-white.
Okay.
But it's still interesting, right? It's not based on like,
Why don't you want to push it all the way?
Why do I want to push it all the way?
Yeah.
So, I mean, I did, right? I mean, that's as far as the technology goes today. So I did that. Like we chose, we looked at all of the data and we made a selection based on the data. But we didn't make the decision based on, was it compatible with life and how did it morphologically look? We went through all the way to the distributor.
What I'm talking about.
Yeah.
Now,
and then we built a spreadsheet.
Oh God, I want to see that spreadsheet so bad.
I promise I will never show anyone that spreadsheet.
I figured that was going to be the answer. Wherever we are today, I have a feeling we're going to be able to read more and more things.
Yep.
One human response that, while I get it because humans are like that, strikes me as very strange. You've said this a few times in a very sort of polite way about looking for extra chromosomes, Down syndrome. Now, when you say, I wouldn't implant an embryo, this is Tom Bilyeu speaking, I would not implant an embryo that had Down syndrome. But then I will get backlash on that because people are pretty fiery about that.
In my family, we have people with Down syndromes, like, in my extended family, right? And so I'm not saying that you should or shouldn't, but it should be your choice. That's I'm a big believer in personal choice.
There's too many morphological characteristics, like this kid is going to be tall or beautiful or whatever that people think, or smarter. Ooh, let's get really weird.
And so people have already said that, right? Like there's a general moratorium in the world, including the United States, on germline editing, meaning making edits that will get passed on to the gene pool. But then you've got certain places like in China at the Beijing Genomics Institute where they are like, we are sequencing everyone and we are looking for genes and gene clusters that can drive intelligence. And so they're very open about that, right? And so it is a really hard thing, right? It's like if we could remove, I'll give you an example, like sickle cell anemia, which affects a percentage of the population, it's one gene edit. It's just one. So if we could eradicate that for all humanity, should we? I mean, I would make the argument yes, but I also don't have sickle cell anemia, right? And so these things go beyond, science in terms of capabilities, but go into not just ethics, but go into philosophy. It's like, I would argue that we have some form of directed evolution as a species, right? We are no longer just hunters and gatherers.
We already have.
We're already doing it, right? It's like when people say that like what Colossal is doing could go too far, or that one of the things we get back, people love to argue whether it's whether we can classify it as a direwolf or not. But what people, which I'm sure we'll get into, but what people don't realize is that like these and what people don't argue is that the technologies that we have that we're now deploying here exist, and they're only going to get better.
Yeah.
And so we have to be thinking about what the applications are for humanity. But I would argue, I always tell people that, we've been doing gene editing for a long time. We've just been really shitty at it, right? Like we've been like crossbreeding stuff forever, like whether it was weed or corn or dogs, like
or
not
who you pick as your spouse.
We have been doing this the old-fashioned way for a long time. Now let's just use data and let's just do it faster and more efficient, where we have a high degree of predictability of the outcome.
Dude, this is inevitable. So I'll put my cards on the table. I have no idea how you're going to react, but we are in a Cold War with China. China is going to do everything that they can to beat us in AI.
I agree.
So we can't play around.
Yeah.
China's going to do everything they can. I saw a video today of a robot dog with a gun on its back and training it to autonomously do its thing.
Yeah.
If we know, so right now, as far as I know, the only place that has done edits on humans is China.
It's China, yeah.
You will often say the person that did that, the doctor that did that, was allegedly punished. I always find the use of the word alleged.
It's interesting. Yeah, it's like you do something that big, it gets out of jail pretty quick.
Yeah, because you have at least running in the back of your mind the possibility that that was sponsored, or at least like, well, if you're doing it.
China's an information state, so I find it hard to believe that people at various degrees of government didn't have the information.
Mm-hmm.
So
it's a very delicate way to put it. Also, I feel like, forget editing for a second, selection of the embryos that you have, I would fully expect people to go as hard in the paint as they possibly can to get the ones that meet whatever characteristics that are desirable.
Can I add something to this? This is crazy. This is from my own personal journey. The morphological embryo that looked the best, based on, this side, like not going all the way up, but just this side, the one that actually looked the best had a genetic defect and was not compatible with life.
Interesting.
So more than likely, our incredible doctor would have put in an embryo that would have led to a miscarriage. Which is emotionally taxing on the family, specifically on the female, and it's also economically taxing. And he wasn't wrong based on the morphological grade. Like there's a grading system, you can look at it, but it was not genetically compatible with life. But that's what would have been implanted.
Right.
If we wouldn't have done these other things. So I do think that data can help us be informed to make, we make more informed decisions with that data, right? It doesn't mean they're always going to be the right decisions, but at least we have the personal choice to make those decisions.
Do you think that there's any of the gray in selecting from unedited but IVF created embryos?
No, I think that's it's like I don't know if have you seen the gallery test, the Grail test, the blood test?
I just got one.
You did?
Literally like a week ago.
You get your results back?
I haven't got my results yet.
That's a little nerve-wracking, isn't it? For me, it is. I do it every year.
I went in because literally as we're recording this, I had skin cancer removed from my face. So I was like, I want to know everything.
But you're one of those people. I'm one of those people. I do the function test, and I do 4 quarterly blood tests. In addition to the function test, which is already insane and incredible and comprehensive as it is. Then I do 4 quarterly blood tests. I do the Grail gallery test, whatever I don't know, Grail.com. It's whatever it is they actually call it. I do all of that because I want the data. But a lot of people they don't want that data. So going back, but that applies. Sometimes it's hard for, I think, people like you and I to understand that. But if you and I, but because we're wired a certain way, but I think that goes the same way with embryo selection. I want the best amount of data to make the best decision, but not everyone does. Some people are like, I just want to trust nature and I just want to trust the universe and trust the chance system.
Of course.
I don't understand it, but they're entitled to it.
I definitely think people should be able to make their own decisions.
Yeah.
I just don't understand the pushback on the selection.
There's been pushback on orchid. And I'm like, here's this incredible woman that developed a technology and just wants to give people more data so that they can make a decision, right?
Say more about this. What is it?
This is the orchid health. This is the orchid test that gives you that risk score. This is the system that we use. But it's like they got pushback when they launched. And I was like, it's just data.
Yeah.
You don't have to use the data. You don't have to take the test. So
can you steel man their argument?
I think that their argument is with more data, you'll make a better decision and you will lead to healthier babies that have longer lives that will also have a longer term impact.
I mean the people that hate it.
Oh, that hate it? I think they think it's a form. It goes back to it's a form of playing God, right? It's like it's eugenics, right? But I mean, I think that selecting for a certain like skin color or hair or eye color, that's eugenics, right? Seeing if your kid has like my gene mutation that has like the issue that I have with my truncation on my Titan gene, that's not eugenics. That's just like I don't want to pass that on. I would argue that things like orchid are a service to humanity. They're not just an individual service because those genes will persist in the gene pool long after you're gone. It's a moral obligation to do this, not just to you and your family and to your child, but to humanity as a whole and because of the ripple effects of the gene pool.
This will get so bizarre. If I'm a kid and my parents chose an option that they thought was cool or kind, or there needs to be a distribution. Like, wait a second, I could have been the embryo that was 3 times smarter. Like, I'm now suing.
Yeah.
That's going to get certainly very bizarre.
It's going to get bizarre. And orchid doesn't test for intelligence, by the way.
Yet.
I mean, I'm not part of that.
And maybe they never will, but that strikes me as one of the things that you should...
To your comment, BGI, and this isn't like some like deep state, like dark web secret. Like they've been, like the CEO is like, yeah, we do that. Like they're very open about it. They're like,
please.
Yeah, they're like, why shouldn't we?
Yeah. That is quite literally my question. Okay, so by what mechanism do we decide what is eugenics and horrible and what is, it's my kid, I want to fine tune a little bit here?
Yeah, I mean, the good news is there's no germline. There's this moratorium on germline editing, and so we don't have to make that decision today.
But let's pick an easy one. So I was just, for whatever reason, this came across my feed, and my wife happens to have a very good friend of hers, mixed race guy, white wife. They have twin daughters, and one looks light-skinned African-American, but not American because she's British, but she looks light-skinned black.
Yep.
And then the other one is ginger, freckles.
Yeah.
Like, how are these two twins? Obviously, they're not monozygotic twins, but twins nonetheless.
Twins, yeah.
And if you have that and you're looking at that and you get to pick, like instead of, 2, or you go through and you want every color of the rainbow or whatever, like, ooh, where do we go?
It is in the realm of sci-fi, right? And it's like leaning on this as it relates to government. What is government's decision in that, right? Like, what is their right? We have a general moratorium on not being able to.
Is that considered germline though? You're just selecting, because that's already like, this is happening right now. I don't know if they were IVF or not.
But I don't think anyone out there has or is surfacing that type of data, like eye color. Like no one's surfacing that type of data. So
people are just like, we're not going to tell you.
Yeah.
I don't think that lasts forever.
I don't think it lasts forever. And one of the things that makes America different than China is our ethics, is what we'll choose to do, right? Like, yeah, I mean, one of the things that, like, Colossal, certified by American Humane Society, the oldest humane organization in the world, we run everything by them, from where our animals live to how we treat them in the lab to every single thing, right? It's hard. It's very, very hard. And if we didn't care about that and we just want to do something in a country like in a Southeastern Asian country and, be like, oh, well, we'll just go kill 100 elephants to do this and we'll have a mammoth in 2 years. Like we choose to do things as an American company in America by American standards, America's ethics, and America's rules. And, not all countries, in this weird, complicated geopolitical world we live in, play by the same rules and play by the same ethical standards. They also look at things. They also look at, like, if you're looking at a lot of the Asian countries, they're looking generational, right? So they're thinking about what is the generational thing, and we're more of a society that looks at today and tomorrow. So I think there's cultural and kind of generational views that go into this that have a very, very big, very like it's like the old adage about like how people steer like a cruise boat, right? If you want to go over there, you make a small turn now and it'll eventually go there. And I think that some countries think like that. And I think that affords them at least a perspective where they don't look at the ethics the same way we do.
Yeah.
We'll get back to the show in a moment, but first, the faster you can get your idea to market, the better your chance of success. And right now, there's a window of opportunity that won't stay open forever. Shopify's AI tools are literally doing the work that used to require entire teams. Need product descriptions? The AI writes them for you. Web design? Choose from hundreds of templates and customize them instantly. Product photos need enhancement? Shopify's AI handles all of that as well. Shopify is not just an e-commerce platform anymore. It's like having a marketing department, design team, and operations expert all working for you 24/7. And that's why Shopify now powers 10% of all e-commerce in the US, from giants like Mattel and Gymshark to solo founders just getting started. So turn your big business idea into reality with Shopify on your side. Sign up for your $1 per month trial and start selling today at shopify.com/impact. Again, go to shopify.com/impact. And now let's get back to the show.
As you look at what is going to be possible, everything has to be done in order. You can't do everything all at once.
You can parallel path a lot of it.
Say more.
Yeah. So in this whole system, right? So while we're doing the computational analysis, we know we're going to edit Asian elephant cells. So we have to get Asian elephant cells, right? We have to get the soup or the medium to grow them and grow them happily. So one of the things that's interesting about I had this conversation last night over drinks with Bob Nelson about P53. So do you know anything about P53?
Yeah, having just had cancer removed from my face, yes.
Yes. So not everyone does, right? And so what's interesting about P53 is us and mice have one copy of it, and in elephants and blue whales, which are harder to study than elephants for the whole breathing thing, they have about 20 times the expression. And what we know about, we don't understand everything about P53, but what we know about it in overexpression is when a mutation occurs, cancer is a mutation, what it does is the cell senesces. It auto terminates itself. It just kills itself, right? So then it doesn't turn into a tumor and then go crazy. And so we know this about blue whales. We know this about elephants specifically. And if you look at the age that elephants live and the body weight that they have, I think it's called Peto's paradox. And you look at this distribution curve, they actually don't get cancer. They get cancer a fraction of what they should, like a minuscule fraction. They basically don't get cancer compared to what we would if we had we have roughly the same lifespan, if not a little bit more now, but had the same body weight and had the same mass. And so we had the same number of cellular divisions. Yeah. And so what's interesting about that is to your parallel path point, we had to figure out if you start editing a cell, looks like a mutation because it is a mutation. It's a forced mutation, looks like cancer. So we had to figure out how to regulate P53. How do you turn it down so you can make the edits?
What animal did you find this on?
In Asian elephants.
Yo.
And then you had to figure out how you turn it back up.
This is in the woolly mammoth experimentation.
Yeah, in the woolly mammoth project. God. So this goes back to your parallel path is while we're doing computational analysis in your data, more data is more data, right? So as we're getting more and we're looking at more of these, as we're running our woolly mice experiment, as we're growing organoids over here, as we're creating induced pluripotent stem cells so that we can create organoids in the first place and then eventually gametes, then we're also working over here on the actual editing of the targets that we made in the woolly mouse of the mammoth equivalents. We're also working on we had to figure out before we could even do that how to regulate P53 and turn it down, but then turn it back up because you don't want super cancer elephants. And then we also have a whole team over here that's working on elephant IVF.
How on earth do you turn P53 down for a bit and then back up?
It's a great question. I don't know. That sounds like someone on our science team that's much smarter than me can tell you that.
That is crazy. So are you turning it back up in the same creature?
In the same cell line, right? Because what you don't want it to do is you want to make sure that you can turn on. It's kind of like the reverse of like immortalization constructs. If you put an immortalization construct so that the cell just keeps dividing, well, eventually it would turn into cancer. You got to eventually, sometimes when you're making lots of edits, take out those immortalization constructs. And so it's a similar kind of thought process on the P53.
That is so.
Yeah.
I don't know what to do with that. I don't know how that's possible. But
it goes back to your core question is like, we're not, if we did all this linearly, we would have animals, it would take us a decade per animal. But, the reason why we have 4 labs, we have 172 scientists, including, people a part of the, National Academy of Sciences, our chief science officer. We have 17 academic partners around the world. We fund 40 postdocs and we have 95 scientific advisors. With all of that, we've tried to pull all that together, and then we've raised a lot of capital, raised roughly $500 million to do this. And you have to do this because you have to build the system. And if you want to do it, and have like all these species back, you have to do it in a parallel pathway.
Okay, why do we want all these species back?
So, a couple of core reasons. Number one, I think that, and I think you probably get this from your background, anytime you look at things as a system versus a point solution is a higher, it's known through like every scientific and every engineering feat, like the Apollo program, that you innovate and build technologies that have far-reaching application than you have any idea, versus a point solution where it's like, oh, I'm going to have a hammer and a nail, and I'm going to I just want this piece of wood to go here, right? Whereas a system model is I want to build a sustainable house that can survive all of the elements, right? So you have to build all of those systems, right? Where you have clean water, get rid of excrement, get rid of all, be able to do air filtration, have power. So it's a very, very different way of looking at things versus like, I just want to solve X. And sometimes I get negative feedback on this when I talk about it as it relates to scientific papers, because I'm a very big believer in looking at the system and documenting it and showing the science over time, but that's not the priority, right? The priority is, can we do this? So anytime you develop a system, and specifically one that has an application to conservation, I think that's interesting and it allows us to innovate. So number one is it's a system model that drives a lot of potential for both support for conservation as well as technologies that could apply for human healthcare, both monetarily and monetized, but also that could also just help humans, right? So that's one reason why I think we do this. A second reason why we do this is I think that, doing things that are hard teach us more. They make us ask harder questions. Like we had a whole conversation about eugenics today. Would we have had that conversation if we weren't talking about gene engineering and pushing gene engineering to the limits because we made a direwolf and a woolly mice? I don't know. And so I think it also helps us from a human condition perspective think bigger and then expands kind of what, like how does it widen our aperture in terms of like what is possible. And then the third thing is kids. Like people like love to just like skirt over this when we're talking, the people that don't love what we're doing love to skirt over this, but every week we get stacks of like letters and drawings and paintings and stuff from teachers and parents and kids saying, my kid wants to go be a scientist, or my kid wants to be a biologist, or my kid and I, like we had a woman from Florida send us a note saying that her classroom won't be quiet except when they're talking about direwolves and woolly mice and mammoths and all this stuff. And they're obsessed with it. And so, I think inspiring the next generation, like the Apollo program and the effects of the Apollo program, I think created, I think it, outside of the success of going to space and opening up space and kind of that, monumental triumph for humanity, I think it changed the human consciousness on a level of what was possible.
Yeah, when I look at gene editing, when I look at particle physics, I feel the same way. That you see these things that just exist. They seem so fundamental. They seem unalterable.
Right.
The process by which they come into being feels like magic.
Yeah.
And then to hear, oh, no, like you can read the DNA, you can go in, you can make edits, you give the virus a little tugboaty thing, it drags it in, it puts it in the right place. Like, it's just like, what? It makes me realize the world does not work the way that I thought it did, and that there are levels to how much I can engineer it.
Yeah, and I think that we're just going to get better at it, right? Like we as a
thing, like this is the trash version.
Yeah, this is that's a good point. We are the trash version, but I do think we're at this point where, you know, over the next 5 to 10 years, we are going to be moving more and more into using AI and big data to and compute to essentially be able to do simulation of design, right? So it's like, how do we, like, we have a tool, this is kind of weird, and this is a trash version of it. We have a tool where, because there's so many different editing modalities, we continually feedback into this model, what works, the efficiency, what off targets were, like unintended consequences happen. We keep feeding back in the model. And so we've got this like this internal software product that we built where it's basically so we can now put in those guides and put in the designs that we're trying to design, and it will recommend what different editing modality which will and predicts what is the highest level of efficiency it thinks that we'll do based on that combination.
Did you guys train the AI yourself?
Yeah. So we trained it all ourselves.
Crazy.
Yeah. Because we're doing a lot of editing.
Do you think that AI will be able to, master might be the wrong word, but will it be able to understand biology in the way that it understands language?
Yes.
How close are we? How long is that going to take?
I think it depends on focus and funding, right? Like, you know, people want it to write, you know, jokes and essay papers, right? It's like if the same amount of effort we're going into AI and training large language models around genomes, you know, I think that would be something that could be done in the next 5 to 10 years.
Whoa.
Yeah.
But
once we do that and we have the ability to do full DNA synthesis, like be able to write everything that we know we need to write with a high degree of confidence that it's going to work, or multiplex edit on a scale where we're making thousands of edits at once with 100% efficiency. Once we're able to do all of those things and do all those things really, really well, then, I mean, I think that the field of synthetic biology, the ability for us to do accelerated directed evolution, the ability for us to, you know, truly guide biology the way we want and not just based on, you know, environmental factors and random chance, completely changes.
Put your sci-fi rudder hat on for a second.
Okay.
So we have the 5 to 10 years of training the model. The model is now, for people that don't know, it's probably worth going into a little bit of AlphaFold, protein folding, how far we've already come on that. But on the other side of that, so we map all this stuff out. Mm-hmm. What starts to happen? Are we like, oh, we take a 25-year-old and we make sure that they can withstand radiation and we send them to Mars? We take a 78-year-old and we make them look 26? Like,
yeah, so all the above. So I think that we will look at being able to engineer things.
Well, first of all, give me protein folding. What makes you believe that this is going to happen?
Why I believe it? Because I think we're getting really we are getting so good at understanding like how a protein, how a specific thing that is made from our body, how its actual shape is and how it binds. So you probably heard about how different blood types and other reasons why certain diseases like COVID, there's different ways that based on how it actually folds, how it fits together. So think of it as like a complex puzzle, right? So it's not a 2-dimensional puzzle, it's a 3-dimensional puzzle. And how you put those things together, they can only bind or fit together certain ways. And it's based on how the protein is folded. We've gotten so good, we as Colossal, we as humanity have gotten so good with Rosetta Fold and AlphaFold and all these tools that we are now understanding the shapes of proteins so that we can predict what it will bind to and how it will bind.
What do the machines look at? Are they looking at a sequence of letters? And they say, oh, this will create a protein that looks like this?
They're looking at a sequence of letters that then will do that prediction. And then I don't know what the functional assays are that they then do it, and the molecular assays are to verify it. but it's highly accurate. So we went through a process with a gene called L-Coral, specifically for which drives size. Or it's known to drive size. It's not the only thing in size. So it's not like, once again, some of these things are multigenic in nature, meaning that we can't just we can't just make the super L-Coral. And then you have like an elephant-sized chihuahua, right? Like you just can't do that. It's a lot of these genes working in concert together that produces some of these phenotypes. But we know for a fact that a truncation in L-Coral, in how it folds, looks at in Great Danes, is different than that in Chihuahuas, for example, right? And we know that drives some percentage of size. We don't know if it's like 100% or 200% or 500% or 20%. But we know it. We do know from looking at mice, from looking at doing a canid analysis, we know from years of scientific research that gene specifically with this truncation creates a larger sized canid. Like we know that, and so when we went to look at the phenotypes for the direwolf, we looked and said, okay, what is the L-Coral variant? Does it match the gray wolf? Does it match a Chihuahua? Does it match a Great Dane? It looked a lot more like a Great Dane than it looked like a wolf or whatnot. So, so we put that variant in, right? And so that truncation in that gene, increase, we have much larger wolves than typical wolves at the size and the age that they are. So we think that, that was, that truncation helped lead to that.
Did you pull the exact Great Dane version of it just to make sure that it would be compatible?
The direwolf version of it. So, because there was debate on this, which, once again, this is an educational opportunity, right? So like there was a paper that came out in 2021, 2022, because direwolves were popular because of Game of Thrones, and they were only able to get, and Beth Shapiro, our chief science officer, along with, another like 30 incredible scientists were on this paper that basically, that the results of the paper, because they only got 0.15x. So they didn't get a full read of the genome. So they only got about 15% of the genome. They didn't get a full pass based on the sample that they had at the time and the technologies that they were employing to do it. They looked at this, and the conclusion that they came to in where a direwolf fell was it was somewhere between a wolf and a jackal. I don't know if you've seen, though, sometimes the results of those discoveries when they make it to media and this was not us, this was years ago, direwolves weren't wolves, they were jackals. That's not what the paper said.
Right.
I was involved, I was running another business. I didn't even know about this at the time. Didn't read the paper. But that's what the headline in a moderately reputable news outlet of today said. And it's like that direwolves were jackals. And so still to this day, so fast forward, Colossal has 500 times, not 500, like 500 times more data about two different variants, or two different samples of direwolves that have 60,000 years of genetic divergence between our two samples.
Whoa.
That's a lot, right? What people don't realize is that there's more genetic divergence between sample A and sample B, that Colossal sequence, than sample A and today. Because that's 12,000 years, right? And so it's like it's very simple, right, to see that. But what people don't realize is, so we took all that data, we went back to that initial author list on that first paper, and we built a phylogenetic tree. And we said, on the phylogenetic tree, where do direwolves fall? And it turns out that they were like everything in life. They were a hybridization of an extinct lineage of canids that were closer to wolves. That doesn't mean that the paper that came out years ago that said but the result was we don't know if they're closer to wolves or jackals. It's inconclusive, but it's somewhere between there. This is just more data so that we know, it skews more towards gray wolf lineage than jackal, right? And that's kind of what everyone thought. But the misguided headlines from 5 years ago, because of Game of Thrones, everyone got so excited, but then they're like, direwolves were like, these are fantasy in the show because they're really jackals, and they're jackals. And that got into like the cultural like knowledge base of the world. And it was just misinformation.
Yeah.
And so we literally get comments where like, these guys don't know what they're doing. They started with the gray wolf. Gray wolves aren't closer.
This is such a weird reaction.
I know.
Look, I fully understand it's what humans do, but
it was fun.
What a strange way to respond in the face of what is possible.
Yeah.
Okay, so why not clone? Why not go grab, like, I get maybe a direwolf, there's nothing where you've got 100% of it, but with things that you could get 100% of the sequence, is cloning better at that point?
Well, you need a living cell to clone, right? Because you're taking a living nucleus and taking it from a living cell and putting it into
That's the only way to clone. You can't clone off of a DNA strand.
No, but what you can do, this goes into the artificial egg, and with mitochondrial, figuring out mitochondrial rejection, and then DNA synthesis. So instead of synthesizing a big block, what if you could synthesize the whole block, right? And so I think eventually we'll get there. It's unclear, but this is where it's funny that we talked about eugenics. I made a joke on a podcast that wasn't well received by the scientific community, which is like, I've never seen the scientific community so eugenicsy about a wolf before, because they all wanted to argue over.
Hilarious.
Because it's true. It's like, if you look at the phylogenetic tree and you look at all life that's ever been, like the tree of life, right? Better known as the tree of life. And you look at all of it, 99.9 to like the nth degree is based on where it lived, when it lived, and what it looked like. That like, we don't have DNA from anything. We don't have, as I mentioned, we don't have DNA from the animals that live today. And so this idea of purity of direwolf is just a psychotic perspective that's just weird.
Okay, well, let me steel man their argument for a second. This is a Ferrari chassis on top of a Toyota engine. How is it not that?
Try to go pet that Toyota engine and see how it works out for you.
Because just the way that
if you look at just their size and where they were located, one, our genetic donor was where we got it from where they're located. So we took gray wolves, which were American gray wolves. We didn't import them, right? So we kind of met that standard. And we looked at, and if you looked at just the, so that kind of fits where they came from. We chose the same wolves from the same place that they came from, right? We didn't import magic wolves, number one. Number two, if you look at kind of the phenotypes, here's what we know about direwolves. This is all we know about direwolves. They were 20 based on the fossil record. I'm going to tell you something else that we didn't know until Colossal did it, but no one seems to care. One is that they were 20 to 25% bigger. They had a larger craniofacial morphology, which implied that they had a stronger bite. And based on their bone density and their bone density specifically in their shoulders and in their legs, most likely, they were heavier, right? So they were a stockier animal. That is what we know. So if we just did that, I would argue that from a functional de-extinction perspective, and the same way that we classify 99.9 literally indefinite species in the tree of life, they would be classified as direwolves. But we didn't. What we also did is we said, oh, this is interesting. Their coats were white. And we know that because we looked at the data and we found in the DNA, but there's no direwolf, there's no frozen direwolves, there's no frozen samples of this. People thought because of a paleo artist rendition of that paper from 5 years ago that they were red, because they were like, well, we got a reddish brown, because they're like, we got to make them look more like, jackals, because they're closer to jackals, because someone read an article that was inaccurate at the time. And even people on that paper will tell you from that paper didn't say they were jackals. I know it because the people that wrote it, because they were like, we wrote it. We're the ones that wasn't in the conclusion of the paper. And so, we actually took it a step further in two categories. One, we found kind of like in the woolly mouse, the hair variant that drives, the hair. And so they have a much thicker hair. I mean, when they were born, they kind of look like baby polar bears. They're amazing. They're like these cute little fluff balls. And then, which there's pictures all over the internet of them. And then if you look at their hair now, it's super thick. There's like waves to it. It's amazing. They almost have this like ridge line to them and like mane across their back, which is amazing. We had no idea of that from the fossil record. We also didn't know they were white. We had only misinformed the public and said that they were red or brown because of a conclusion of a paper that the paper didn't come to that conclusion anyway 5 years ago. And so I would argue that not only are they direwolves, based on how 99.9% of every species is currently classified on the planet, but they're even more direwolves than anyone could predict based solely on those phenotypes, because we identified things in the DNA.
And so you guys, how many like DNA blocks?
So we edited.
whatever we
It's
call
a great
them,
question.
Did you put in?
So we edited 14 genes with 20 edits.
Okay. And starting with a gray wolf.
Starting with the gray wolf. This is our genetic donor because they're 99.5% the same genetically.
Got it.
Comparable to what Asian elephant is to mammoth.
Okay.
And most people don't realize this, but Asian elephants are closer related to mammoths than Asian elephants are to African elephants.
How do we not end up in a Jurassic Park scenario where we made something and it has an unintended consequence in the ecosystem?
Well, it's a great question. And for all these species, we go through, like, people think about, and I don't think that was the exact premise of Jurassic Park because they weren't rewilding the Jurassic Parks, right? Like they did not have an ecosystem restoration or a conservation subplot that, unless that just got like, on the cutting room floor. But so I don't think it's exactly the Jurassic Park scenario. But to the ecosystem restoration unintended consequences is we have to measure all this stuff, right? Anybody that tells you like, hey, you can introduce this species back here, will have zero unintended consequences or will have this intended consequence is just wrong. But the beautiful thing about rewilding is that Colossal, people kind of, I think, once again, give us too much credit. They're like, oh, they're just going to rewild them. It's like, we have a foundation that we raised $50 million for, and we have 48 conservation partners, including some of the top rewilding partners in the world. Specifically on the Direwolf project, we actually work with some of the gentlemen that ran the Yellowstone rewilding program for gray wolves back into Yellowstone. So we, if the government, indigenous people groups, and ecologists and everyone wants to rewild direwolves at some point, we would work with them to do that, right? Like we built a rewilding plan because I think anytime you do this, you should at least be like, what are the possibilities, right? So you should think about that. But they live on a 2,000 acre secure, expansive ecological preserve that are monitored 24/7. They've got 10 that's certified by American Humane Society. They've got 10 full-time people to attend to them, security. They've got storm shelters and we built the animal hospital in case anything happens to them that we could treat them right there. They don't have to it's not like calling 911 and then having to take them to the hospital. We have a hospital and a full-time vet there.
Are you guys going to make more direwolves?
So we are going to make 3 to 5 more. So we want to have a pack size of roughly 5 to 8 because...
Will you impregnate the ones that you have?
No, we'll engineer them. So we're going to engineer them.
And they'll be birthed by a gray wolf or by a dog?
No, by a domestic dog.
That's crazy. What kind of dog?
So they're just large hound mixes.
Is that dog like, what just came out of me? Or do they just
No, they nurse them and
glad
everything.
You're here.
You get the colostrum from it and everything. They nurse them and take care of them. It's great.
So to them, it's like, I just have weird looking kids.
Yeah, or super cool kids. Depends on how you want to look at it. But then all of our, and then we work. This to me is kind of fun. Is that because we work with American Humane Society so well, all of our dog mothers, our surrogates, get adopted through an anonymous adoption program to their forever home. So that's kind of fun. There's people out there that have, moms of direwolves.
But you don't want them to know for some reason.
I feel like that's like a spectacle of the animal, right? Like, not saying that anyone do that. Like, I think that if people I know adopted them, they'd probably be like, oh, that's cool. And they may tell their friends, but who knows? I mean, people are, we're all weird in our own ways. Someone could try to exploit that, right? And be like, oh, this is the mother, like in Game of Thrones, like Mother of Dragons, this is the mother of direwolves. And it's like, I don't think that's good for the animals.
Oh man, this
but
is so intriguing.
What people sometimes don't think about is like, if my job was to raise money and hire an awesome team and let them just do stuff in the lab, this job would be very boring and easy. What's interesting about this job is that we have conversations like this, right? We go meet with the government, we go meet with indigenous people leaders. We work with, okay, how do we adopt our surrogate moms, right? Like there's so many nuances to how we think about this that's what sometimes is hard, that unless you say, when we launched the Direwolf story, we worked very closely with a handful of people, Rolling Stone, Time Magazine, the New Yorker, and others, even though the New Yorker broke embargo and kind of screwed us for a while, which was very, very painful week for us because we didn't have our website up. We didn't have our scientific paper hadn't been submitted.
Did they apologize or did they do it on purpose?
Well, they definitely did it on purpose because it was in print. You don't like, it's not like someone like spilled coffee and hit the publish button, right? They're like, oh yeah, that looks good in InDesign or whatever. And that's going to the printer and that's going to newsstands tomorrow. So that doesn't feel like an accident.
Yeah.
So they have not publicly apologized, but we'll just never work with them again. So it's fine.
Yeah. Interesting. Okay. Let's bring this all back to humans.
Okay.
So you've got Brian Johnson, the Don't Die movement.
Yeah. I know
longevity,
Brian.
escape velocity. How
Peter
plausible.
Diamandis. You actually have a lot of people here in LA.
Yeah. No,
Yeah.
Peter I know well.
Yeah.
Brian I know well-ish.
And I don't think Brian gets enough credit because like people like, you know, we live in this like armchair Twitter X of people where they just want to criticize. They don't want to do anything, right? Like what's that famous quote that's like, you know, that you know, people are either on the field or in the stands, and there's a lot more people in the stands bitching with people on the field.
A lot.
Yeah. And like, you know, I'm not going to do everything that Brian does because that is insanely hard lifestyle, but I'm glad that someone's doing it. Like, so that's the way I look at it, right? It's like, Brian's not hurting you.
Yeah, I don't understand.
Brian's not, but there's this weird backlash towards Brian. Wow. I don't agree with everything he's doing, but he's not like saying, you know, hey, you have to do this. You have to do this. You know, he's not showing up with like ski mask and like making people do red light therapy. And so it's.
So that I kind of would get behind that.
I would too.
Let's go, Brian. New business model for you.
I love red light therapy. But like, I think that it's unfortunate when people like Brian get backlash. It's like, this guy is taking biomarkers to an extreme, and he is running an experiment that someone in science we should be lucky. We should be thanking him that he's doing this.
Yeah. Kick off as much data as you can.
And I'm not saying I agree with all of his the results from it, but you know, if you sit down with Brian, which sounds like you have, he'll tell you like the number one thing is sleep. Like the first is like, you don't even have to get weird on like your eating schedule or workout schedule. He's like, you really need, he said the number one thing to me is like what your resting heart rate is and variable heart rate is going into sleep in that transition mode to sleep.
Yeah, that makes a lot of sense. Do you think we will actually hit longevity, escape velocity, where science adds a year of life for every year I live?
Yes.
How soon?
In the next 20 years. 15 years. Yeah.
What are the sequence of breakthroughs that we need to get there?
I think the biggest thing that we need is, so there's been a lot of stuff on cleansing blood, I
didn't see that coming.
Yeah. So this is a newer
like
thing.
super kidneys?
No, you remember the Silicon Valley Blood Boy episodes?
Yeah.
Where they'd like circulate the blood.
Bro, if that was real,
get
your boy hooked up.
So get ready. So there is not that service, but there is a service that's not currently available in the United States. I have not done it, and nor do I endorse it because I haven't done the research on it.
Sure.
But Gary Brecka's talked about it. A handful of other people have talked about it. But where they basically it's like a super dialysis machine, right? So it's a transfusion, takes all your blood out, runs it through a system that's basically like this super mesh, and it takes out like precancerous tumors, old cells, just all this stuff, and then it pumps it back into your body. And it's supposedly like the blood that goes back into you is like the equivalent of like a 26-year-old. So that's
Do you know what that's called?
No, we can find it. We just called.
My wife did something like that.
Peter Diamandis knows it too. I forgot. So I think you're gonna have like the cleansing side of that side, right?
Yeah.
And then I think what you have, and so I think that's here today. Another thing that's here today, if you look at like the big causes of death in America, is like you have heart disease, you have diabetes. Many people think that Alzheimer's and dementia are basically type 3 diabetes and just led from inflammation, right? Which is diet, and a lot of things can be driven by that. But if you look at things like cholesterol, we have medications now that you can absolutely lower your cholesterol and not have heart buildup. So Gary and I have a site. I'm learning more. Gary's got really good ideas, but you know, my LDL is in the 30s, which is, and what they've shown is anything under 60, you start to reverse any plaque or any buildup in any of your veins and arteries. And
you got there by taking something.
Yeah, I take a Repatha, which is made by Amgen.
That's an injection?
An injection. Yeah.
Okay.
And so you have things like that. Then you've got things like metformin, right? And you've got other things to lower your A1C. So I would argue that we have roughly defeated diabetes and major forms of heart disease today, right? Like I truly believe that. Like, you know, if you don't eat sugar, if you eat a moderately healthy lifestyle and you take certain therapeutics, we have done that. I think the GLP-1s, some people hate the GLP-1s, but I think the GLP-1s are really, really interesting. They're starting to show that because you have these GLP-1 receptors not only in your brain, but all of your body. You're starting to see things like kidney function improving, muscle function and heart improving from this. And these are all non-BMI related studies. So there are people that think that GLP-1s will lead to lower Alzheimer and dementia, but those are
So do you think the punchline is going to be a bunch of exogenous stuff, or do you think the punchline is going to be AI reads your genome, we get very good at this stuff that you're doing?
I think that the punchline for longevity escape velocity, which is 1-to-1, is a combination of therapeutics. And then I think eventually, kind of like the function blood test or others, you will get a combination. So instead of taking like a Repatha shot, a GLP-1 shot, whatever the frequency is and whatever the doses is, you take your metformin. I think you'll have a single like shot. I think it'll probably be administered via shot versus orally.
That's really interesting.
So I think you'll have like a combo shot that is like your longevity shot that keeps all your biomarkers, that gives you like Brian Johnson level biomarkers. So I think you'll have that. And I think you'll have that before we have, the point that we are, doing full kind of like AI. And I think AI could be applicable to that, but I think eventually we have AI that, tells us like, hey, here's XYZ, edits to your body that is
because you're close enough to that problem to know how hard that's going to be.
Yeah, it's really hard. But I mean, there's people out there like, I mentioned Bob Nelson, he's got numerous companies, including like Altos Labs. George has Rejuvenate Bio that's doing insanely cool stuff. And there's biotech companies right now that are in preclinical trials and some in clinical trials that have, potential, like massive potential for longevity.
It's interesting.
Because remember, longevity, I think that there is I think that there is and I think AI potentially could help solve this, but there is a very large drop off in that transitional data between like the world's experts. So if you take like the world's expert in like epigenetics or the world's expert in genome engineering, like George, right? And he dies, you can go read all his research and you can go, but unless you've trained an AI to think like George, be creative like George, have every thought that George has had, then you're going to lose, even if you have the next best geneticist in the world. They're not like, there's still like this generational loss of data that occurs, right? Because there's problems that you probably think of or I think of that we don't write down. Doesn't mean that on some like background thread they're not running all the time and you're not thinking about them. But you probably just haven't like, I'm going to go solve this problem, or I'm interested in this and I've accumulated data based on this interest. But you haven't like put that into something that if you were gone, that I or someone else could go read. And so I think AI can help with that. But I think that if we can increase some lifespan and increase, even if we don't get to full longevity escape velocity, I think that coupled with AI, that coupled with health span so that you are more productive to society longer, you eventually get to the point that it's where it's just immortality.
What are the roadblocks that you see coming with what you're studying right now? Like when I look at AI, I say, okay, well, if there is no computation problem, meaning we have enough chips, if there's no energy problem, then I don't see why we don't get to ASI. Do you see, like, are there similar things like that with what you do where you're like, well, we'd first have to be able to solve for this problem?
So for us, with what we are trying to do at Colossal, there's no like FTL problem, right? Like, it's like, we don't have to like solve faster. Like, we're not like to get to our Alpha Centauris, we don't have to solve faster than light travel. It takes us a longer time and it costs more money, right? So ours is an efficiency. Ours is more of an innovation play than an invention play, right? Like we are trying to innovate these technologies. Now, that doesn't mean we're not trying to invent new things along the way. So we are doing some discovery, but our bank is that AI and some of these other access to compute will accelerate the efficiency of these technologies because we are doing it right now on a rudimentary scale. And when we started the business, people were making one edit at a time. Our thylacine project, we've made 300 edits in the cell line. We haven't taken that to term yet because we haven't solved the cloning, the somatic cell nucleotransfer process in marsupials yet. We have a team that's working on it.
Why is it different?
It's so once all of these non-model species are unique. So for example, this outer shell in a marsupial, because you'd think giant big elephant, right, or human, right, and then you see these like little bitty Dunart, and you'd probably think, well, it's probably got the weakest little cell ever. It's got this Zonapalooza, this outer shell.
Zonapalooza,
which is insane.
That's the literal name?
Yeah, that's the literal name. So we have created this laser assisted system for drilling in using a laser in so that we can extract DNA. We're now doing it with computers and robotics and AI. It's pretty and computer vision. It's pretty awesome. But we hit it with the laser, doesn't move the outer shell.
Whoa.
Doesn't move it. We go up to like 11 or whatever the highest dial is, and then some, and the cell just blows up because you just like hit it with like the Death Star, right? And then if you use, and the reason you use that, because if you use like a needle, if you're going in and you're jamming the needle, it's like you are damaging the DNA in that, right? It's like the fact that like Dolly even worked with like all the blunt instruments is magic in itself, right? I don't think the team at Edinburgh and the team that did Dolly gets enough credit. Like that is a miracle in itself. Like people don't realize that. They're just like, oh, they just moved a cell. But given that they were like they're like flying using like, nails and hammers and wrenches. Like it was incredible what they did. And so now it's come a long way. But so for specifically that, we actually had to invent so a lot of times we'll even make our own tech. So we actually make our own needles, in many cases our own glass needles. So we had to make it out of quartz so that we could vibrate it at the right frequency, so that the vibration, because the glass couldn't take it, but the quartz could because it was hard enough. And then that quartz needle at that vibration could pierce into it because the laser wasn't strong enough. And when we dialed it up too much, it just destroyed the cell.
That is fascinating.
But see, those are the weird things that we have to solve when people are like, oh, it's just the most genetically modified animal on the planet. And I was like, yeah, but we had to do a bunch of other stuff.
What do you think is the upper limit of the number of edits you can make?
Without DNA synthesis, without just synthesizing it? And you mean through multiplex editing?
Yes.
Thousands. Maybe tens of thousands.
Whoa.
That's all you really need. Because you're doing target. I mean, I would argue though, once again, this is coming from a software perspective. I'll get shit about this from a biologist, but the smarter you are on computational analysis, the more you know, the less edits you make. Because every edit you make is going to have some level of risk.
Because things are redundant or they're not necessary to the final form.
Yeah, and the thing that people don't realize, like when you get a genome sequence of all these letters, right, and you run it through, the reason why you need so much coverage, we talked about like, I think we ended up having 13 or 14x coverage. That means that we had 13 or 14 full reads of the genome for the Direwolves. Whereas before they had 0.15x, they didn't have even a full one read. The reason why you need that is because, and you need to have at least probably about 6 or 8x, I think, to do de-extinction properly, because the machines aren't perfect. So, and the DNA is degraded. So it's giving you at every single site where it's saying this is a XYZ letter, right? This is a C or a G or whatever. When it's doing that, it says these, it's giving you a probability score that this is a C. It actually isn't 100%. So they're like, oh, it's like 50%, 99%. So the more you do, then the higher probability is that you get that, right? And so even when we as humanity get to the point that you can synthesize a full genome, and this is where, not to argue the weird eugenics points again about like what makes a mammoth a mammoth, even if you get to an end-to-end mammoth, there is no end-to-end mammoth genome. So right now we're doing an assembly of a mammoth genome, building a reference genome, but we're using 60 genomes that we're putting together, right? And so then you've got to go look at that, and then you've got to go say, so even if you synthesize all of that, you're still going to have holes. So we announced in October of last year that we have a 99.9% complete genome for the Tasmanian tiger or thylacine, which is amazing.
How are you picking what animals you do in what order?
Well,
like a thylacine. What the?
Yeah.
What?
So
it sounds like a period in time. It does not sound like an animal.
Yeah, it's awesome.
There was a Jurassic and
but
the
you've
thylacine.
seen a thylacine, right?
I think so.
Can we show you?
Yeah, pull it up.
Yeah, can you?
Let's see.
It's amazing. It's
Yeah,
like
I
a
have seen this
zebra.
before. It looks fake.
It's like a zebra, wolf,
It's
kangaroo.
like a kid was drawing and had a seizure halfway through. That's
Yeah.
what that looks like.
It's awesome. And there's videos of them and everything.
Are they going extinct or are
No,
they
they went
extinct?
extinct in 1936.
Yo.
So they're awesome. They're
Okay, but why? Why this one?
Jurassic.
Oh, so specifically why the thylacine is, we hunted it to extinction. So the Australian government put a bounty on its head and actually paid farmers and hunters to kill the thylacine.
Doesn't that tell you this thing's a pain in the ass?
No, they got a bad rap. The sheep farmers, which were stealing each other's sheep, killing each other's sheep for competition, blame the thylacine. There's no data. Like I've spent a ton of time in Tasmania. I spent a ton of time with all the thylacine experts and the researchers. There's no data that shows that they would ever attack a sheep or eat anything that size. They actually killed mostly kind of those mezzanine marsupials like Tasmanian devils and whatnot. So they were
Now, is this a marsupial?
yes. So
Huh.
yeah, and like the wombat, it's got a backwards pouch. So if you think of most marsupials have a pouch like up front and
like a
the
kangaroo.
joey's in it, but it suggests that this was and there are thylacine dens that have been found. So it was a burrowing animal. So it actually burrowed, and if the pouch was facing forward, it would fill it full of dirt and kill it, just like the wombat.
That's so
Yeah.
cool.
Yeah, it's super cool. But so we picked that species because one, we as humanity made it go extinct. Two, there's great DNA. This is not an ancient. We had to go build the genomes, but this is a genetic engineering feat, right? So there's 70 million years of genetic divergence between a fat-tailed Dunart which is like a marsupial mouse, and we're turning it into a marsupial wolf, which is crazy.
That is crazy.
But it's amazing. Yeah. So, but
Wow.
But once again, going back to
So
software.
you pull just sort of from what's going to be the most fascinating. What do we have connections to that we could rebuild?
So what is possible, right? Is there a reason to it? In this case, we removed it. There's not been another apex predator that has replaced the thylacine in lower Australia or Tasmania. And we know
Is that creating problems?
Yeah, so there's a concept called tropic downgrading where you basically have it's a ripple effect, right? So you have this entire ripple effect on the ecosystem when you have a missing predator. We've seen this in Yellowstone, we've seen this all over the world. And so what's interesting for the thylacine is that have you seen this? We shouldn't look it up because it's awful to look at. But have you seen the facial tumor disease?
No, I've heard you talk about it. I very aggressively resisted the
Don't
urge to
look it up.
up.
It's
Yeah.
rough. It's like it's bad. But if
Don't you dare. I could see him over
yeah.
there typing already. Nope,
it's so bad.
not here for that.
It's like out of a horror movie, right?
I'm going to have my producer Drew arrested.
Yeah, it's bad. But what's interesting is that if you look at predators, they have some internal calculus of energy expenditure, right? So not every pursuit of a kill results in a kill. And we see this in Africa with cheetahs and with big cats. If they go so long without making a kill, they're so unsuccessful after making a kill, eventually they're too tired to pursue it again and they die, right? So there is some internal biology, model that they use to decide whether they're going to go after a kill. But what's interesting is, so they typically pick the young, the old, and the sick and weak. And so this goes back to the survival of the fittest. This is great from a genes perspective. And so there's been people like Dr. Andrew Pask and others that have said that, if the thylacines were here, there would still be probably the facial tumor disease, but it'd be a lot more controlled because if you see a Tasmanian devil with facial tumor disease, it's like walking, it's stumbling, like it can't see well. So it kind of attacks everything. That's how it transmits its disease so much, because when they're eating, they'll actually attack each other because they're pretty aggressive when they're in these like swarm eating.
Right. Woof. Have you read the book 1493?
No.
Oh, man. Dude, you've got to read it. It is all about how humans have transformed the earth in ways that they were never even aware of.
Yeah.
Like, I am almost certain the following is true. Earthworms are not indigenous to North America.
That's amazing.
Oh, I was like, what?
What?
And they were brought over here because they would use dirt as like a ballast in ships. And so then when they got here and they unloaded everything, or when they loaded things up, so they would bring the dirt over because they knew they were going to load a bunch of stuff up. So when they were about to load up, they would dump all the dirt, and in the dirt were a bunch of worms, and they have spread all across North America.
That's crazy.
And just completely changed the soil and all of that. Same things with potatoes. Like potatoes are from
Yeah, we're bad at this. We like
crazy.
you probably heard me talk about like the cane toads. Like we introduced this to Australia and they're killing all the marsupials.
Yeah.
Because the marsupials didn't evolve next to them, so they're eating them and dying. Yeah.
Is that a big driver for you guys? Just like
We think
ecological balance?
Yeah, so we think it's interesting, right? So like, in the cane toad project, we have made a single edit, so one edit, and it confers 5,000 times resistance to cane toad toxin, which is super cool, right? Because then you can make like one gene edit, you can make a handful of these super quolls, which is what's eating them and dying. And then other animals are eating them too and dying, but not as much as the quolls because frogs and toads are primarily their diet. Well, then these super quolls can get that population under check. And then the quolls can live, and then it can also have this halo effect of getting rid of the cane toad. You can have even a bigger halo effect of protecting other marsupials.
Have you come across any cool things like that in humans? Like you could have an increased tolerance for plastic, which I know you have a great punchline for, or because we've got microplastic problems, we've got radiation.
Yeah, I think radiation and cancer suppression are two really interesting areas that, I think that data that we see from some of our work and that we see other people working on, I think that could be minor changes. I think we'll have a cancer vaccine in the next, not we as Colossal, but I think humanity will have a cancer vaccine.
Why a vaccine?
Because it's about 200 different types of diseases. Like cancer isn't one thing. It's about 200.
Why does making it a vaccine work?
Because then it just trains your body to go attack it.
Interesting. Huh.
There's a lot of people obviously focusing on cancer, which is great. But, going back to like longevity, escape velocity, that and, Alzheimer's, unless we can see if Alzheimer's and dementia is directly connected to environmental accelerants / inflammation. So
talk to me about this enzymatic breakdown process. The company's called Break or Breaking?
Breaking. Yeah. Breaking.com.
This is...
It's cool. It's great. Yeah. So Sukanya and Bhaskar and their incredible team at the Wyss Institute actually found this microbe that when put in with only adding salt water.
Naturally occurring?
Naturally occurring. What? Naturally occurring. It would break down and visibly degrade, at the time we didn't know why, any type of plastic that was in there. So if you had a vial of salt water and you put in and you put the enzyme in or put the microbe in that creates this enzyme and you put nylon, fishing nets, anything in there, right? I guess it's mostly fishing nets, any other types of industrial plastics at different rates, but it would take plastics that have never degraded or only degraded a little bit, or would degrade over 800 years and, in a couple of years, completely degrade them. And what it was doing was it's breaking the chemical bonds. That's why I named it breaking. It actually breaks the chemical bonds in plastic.
So it turns plastic into just normal biomass.
That is,
it's
bananas. Awesome. It's really, really cool. And so what we did is we one of the things that's been this halo effect of Colossal that's been pretty interesting is that because we have a computational biology core, right, and software, and we know how to look at genomes and this type of data, and because we have genome engineering capabilities, a lot of researchers are bringing us really cool projects where they need AI, computational analysis, and genome engineering. And so they came to us and said, hey, we have this microbe. We think it does this. Can we do an analysis? So we did a lot of analysis work and we looked at it and we found, it's creating this enzyme. It's actually a couple of things working together to create this specific enzyme that breaks down, and everything we threw at it broke down. So then we're like, how do we make it hungrier? How do we make it where it exudes more of that enzyme? That's a genetic engineering challenge. That's not a that's not just a directed evolution challenge, right? Like we're not going to be like breeding this, right? We're going to be accelerating it through synthetic biology and actually editing the genes that make some of those enzymes.
That sounds like a huge breakthrough. Is it a big breakthrough?
Yeah, it is. I mean, right now, our goal is, my goal is to get it to 24 hours. They can break down any plastic based on the amount of distributed surface area that you need, which is really interesting because a lot of the plastic degradation tools out there are just making smaller plastics. They're not actually disintegrating the plastics, number one. They're also not, a lot of them are having they have to be heated or pre-treated with the chemical. Some of those chemicals are worse than plastic.
Great.
Yeah, so it's not helpful. And so our thought process was, so we did this, we incubated it, we got it where we wanted it to be. Now it was 22 months. Now it's about 18 months. But our goal is to get it to 24 hours.
That's still
it's amazing.
Massive.
It's
if
truly
it was
amazing.
800 years to 2 years, is already
it's huge. And we currently have 14. It's either 14 or 16. I should know this because we just had a board meeting on it. But 14 or 16 pilots. So we're working with, I can't say the names of them. I mean, I could, but I don't think I'm supposed to. But screening things that come through water, and then treating it. Number one. Another area is textiles. Like a lot of the stuff that we make, especially like in fast fashion and others, has there's a lot of plastics in our clothing that most people don't realize.
Yeah. That's really starting to make my radar now.
Yeah.
That one feels like it's massive.
And we're gonna rate. It's going well. So like I think about these companies in kind of like, 3 categories, right? Like, is there a general desire for them in the world, right? Is it solving a use case, and is there a general desire? Because those can be the same or those can be different, right? I think everyone generally agrees that loss of biodiversity is bad, and everyone generally agrees that the accumulation of plastic, everything from the oceans to our bodies, is bad, right? And I think that everyone agrees that doing really cool things and inspiring kids for science, and in both those cases do it right, then those things are good. And so we, in both of these projects or companies, we see those all science, yes. Well, then the science has got to work, right? So like we have to be able to create a woolly mouse. We have to be able to create a direwolf. We have to be able to create, degrade plastic from 800 years to 2 years. And then it's really just the business modeling side, right? And so, I've kind of fallen, coming from software and not being a biologist at all and kind of being good enough at learning it, I've just fallen in love with this idea that the combination of AI, compute, and synthetic biology, we can solve a lot of really cool challenges. And I think we can do it in cool and interesting ways that gets people excited, right? Like if you have a plastic degradation company, that's awesome. If you then take the plastic degradation company, work in a bunch of textiles behind the scenes, that's awesome. But then if you do that and you bring in like, people, from like NOAA or from like Jacques Cousteau's foundation, or celebrities that care about the ocean, then you're going to bring awareness to science and these problems. And so, one of the things that I think that we do with our companies, which is interesting, is like we're solving really hard things, but we also try to do it in a fun, flashy way that gets people excited.
What's the coolest challenge you want to solve?
With these companies or ever?
Let's go with ever.
I think it'd be really interesting. So I don't want to be in the therapeutic space. I think there's enough people in the therapeutic space, but I would like to, if I could have 2 solutions, is I would like to be able to engineer plants into doing whatever we want. George and I joke about this treehouse concept of like, not to get too weird and hippie-ish, but it's like, why can't a tree grow in the form of a dwelling, and why can't it like, we weren't on mushrooms when we did this is true, but it's like, but then why can't it have bioluminescent fungi and it light everything, and like, it would be like the ultimate kind of like cleansing for the environment, right? Because you see redwoods that live in all kinds of environments. So I'd love to be able to engineer plants in a way that like we're not just like chopping down forests, but that like the plants could build our cities. That's like the weirdest, I'm not working on that, but it's a really weird thing. That's one. 2 is I'm very interested in the oceans. We're not working on this either right now, but I think that there is a huge opportunity. If we look at, the most stable patterns of weather globally and where life persists consistently, it's somewhere between 30 and 70 feet under the water, and it's pretty stable there, regardless of like the ocean churn above it. So if you could build tools and technologies to make coral more resilient and through synthetic biology, not by just microfragmentation, then all of it grow faster, but it's still going to die. So you have to do genetically modified corals. If you do that and you build closed systems for underwater living, I think that's really interesting.
That's very interesting.
Because I mean, if you look at the ocean and you look at the surface of the earth, like you could build. I mean, what is the economic value of the California coast? $1 trillion? I don't know. It's a lot, right? If you owned all of it, well, there's no wildfires 30 feet under. You have to build closed systems. So the same technologies that you're developing without needing major heat and cooling inversions and radiation tolerance that you're spending a fortune on cost a kilogram to put to space, you could apply those same technologies to Earth and work on underwater cities.
Do you think that's going to be something that happens in our lifetime?
I mean, I think that we're going to achieve longevity escape velocity. So the answer to any of those questions is yes. Because our lifetime's indefinite.
Yes. Oh, God. I hope that really comes to pass.
Yeah.
My man, I cannot thank you enough. Where can people follow you?
I'm just on Twitter or X, just at Ben Lamm.
There it is, 2 M's. All right, everybody. If you haven't already, be sure to subscribe. And until next time, my friends, be legendary. Take care. Peace.
If you like this conversation, check out this episode to learn more. Due to AI and a changing global order, the world is in the middle of the greatest period of change ever. But because we're in the middle of it, it is nearly impossible for us to accurately see what's going on. We are in the fog of war. While the world panics over job loss and killer